Hello, biology world. Purpose of this video is to give those students who were absent today an opportunity to get the 12-2 class notes done. Also to give those people that were writing by hand an opportunity to finish up any of the notes that they missed. It is my lunch hour, and I do apologize in advance. I will be eating while you guys are writing or watching. Remember, Cornell note style, question on the left, answer on the right. And for this, for those of you doing a Google Doc, you are going to need 12 rows and 2 columns. And we start the numbering with 25. We do not start the numbering with number 1. Okay, and remember, for those of you that are going to mute the volume to listen to your own music or just listen to something other than Paulus, I will point at the camera when I'm ready to go to the next slide. Of course, if you're still writing or typing at that time, that would be when you would pause the video. Here we go. 12-2 talks about chromosomes and DNA, and then DNA replication. Prokaryotic cells are single-celled organisms. Bacteria, some pond creatures are prokaryotes. They're simple creatures. They don't have a nucleus. Therefore, the DNA just floats around in the cytoplasm. Chromosomes, they have one long segment of DNA that coils into one chromosome. Okay, so those are prokaryotes. More complex organisms, us, who are multicellular, are called eukaryotes. Eukaryotes, most of us have 1,000 times amount of DNA as prokaryotes do because we're more complicated. We have more characteristics. And our DNA is found inside of the cell's nucleus. It's protected. It's organized. All right, here we go. Moving ahead. Number 28. So vocabulary, eukaryotic chromosomes, DNA and protein, tightly packed to form chromatin. That's one of the items on your study guide. Chromatin is DNA wrapped around proteins called histones. And then a nucleosome is about, from the diagram, if I remember correctly, it's about 8 histone proteins with the DNA wrapped around them. From smallest to largest, you're going to need this for the study guide. DNA, chromatin, coils. No, pardon me. DNA, chromatin, nucleosomes, coils, supercoils, chromosome, from smallest to largest. If you're curious, you're probably not. Homemade pinto beans. I season them. I make them gallons at a time and freeze them with some leftover. We didn't do traditional Thanksgiving dinner on Thanksgiving. I made fried chicken, as I like to do. So we had our turkey a couple nights ago with homemade turkey gravy. Beans and gravy. Yummy. All right, getting ready to move ahead. Okay, so this is what it looks like. So this is DNA. These are the histone proteins. DNA wrapped around the histone. So this would be like chromatin here. A nucleosome is this entire ball of histone proteins. Just like when you're making a ball of string, you have to have something in the middle to wrap it around. That's what the histones serve as. So this circle here is a coil. And then this is a coil, this is a coil, this is a coil. So these are supercoils. And then, of course, supercoils form the chromosome. So smallest to largest, DNA, nucleosome, coil, supercoil, chromosome. Okay, moving ahead. Okay. Please use this as an opportunity to practice with your notes rather than just fast forwarding. How many DNA molecules do prokaryotes have? How many chromosomes do prokaryotes have? Eukaryotes, of course, have a lot more. If you're not sure, take a look back at number 25 and 26. One piece of DNA, one chromosome. Well, one piece of DNA, one chromosome for prokaryotes. Okay, eukaryotic chromosomes contain DNA and protein called. So what is the DNA wrapped around the protein called? Take a look back at number 28. Chromatin. Proteins are called histone proteins. These DNA and the protein form nucleosomes, which pack together to form a thick fiber. I actually cut that out of your notes. Okay, moving ahead. Here we go. It's a DNA replication in prokaryotes. It's a lot simpler than it is in eukaryotes. One strand of DNA has the information needed to make the other half by base pairing. Let's make a little bit more sense at the end of these notes. And it starts at a single location. Okay, totally different than eukaryotes. Eukaryotes, there's a lot more DNA. Therefore, it can't just start at one location and go in one direction. Eukaryotic DNA starts at hundreds of locations and it goes in both directions to make it as fast as possible. And the location at which replication takes place is the replication fork. All right, getting ready to move ahead. The importance of replication. So cells are just like organisms. They grow old and they die. And the jobs that they do, they can no longer do. So cells need to divide or replicate to replace the cells that die. So replication ensures that each resulting cell, the new cell, the replicated cell, will have a complete set of DNA when the cell divides. And of course, the DNA is the instructions for the cell. So the DNA replication has to be accurate as well. And as you learned in the Crash Course movie, DNA replication for, why is my hat all jacked up? There we go. Within humans is very, very accurate. I think it was 1 in 10 trillion base pairs. Does a mistake get made? Something like that. Replication details. So the DNA molecule separates into 2 strands. Remember the helicase enzyme separates breaks the weak hydrogen bonds and separates the 2 molecules. Then those strands are used as a template. DNA polymerase brings in new nucleotide bases and uses base pairing rules to make a new complementary strand off of this one. Over here, this was the original or old strand. DNA polymerase brings in new nucleotides to make the new complementary strand, and you end up with 2 double-stranded molecules that are identical after it works. So new and complementary mean the same thing in terms of the strands. Old or original mean the same thing in terms of the strands. All right, getting ready to move ahead. Okay, so you had to, no, you will have to fill this out in class, although if you're watching the video after you took the notes, you did fill this out. Okay, so the way that you know that this is the new strand is if you trace this around, you can see this is the location where new nucleotides are being added. Therefore, this is the new strand. And of course, this one is the template. You can see these bases are here. DNA polymerase, which is this circle, is going to be bringing in new bases. Okay, so new strand, original strand. These are nitrogen bases, which make the steps or the rungs of the ladder. Notice that the growth is, well, the DNA replication is happening in both directions. Okay, it only happens in one direction for prokaryotes. Location, where helicase unzips the molecule and new nucleotides, or pardon me, new nitrogen bases are being added, is referred to as the replication fork. Same thing over here, replication fork. Eukaryotes, this is happening in hundreds of locations simultaneously. And of course, this bubble-looking structure is DNA DNA polymerase, which not only brings in the nitrogen bases, but it also proofreads the work that has been done after it has been done. Okay, moving ahead. All right. All right, so the replication enzyme is DNA polymerase. Again, don't get that mixed up with the helicase enzyme. The helicase enzyme is the enzyme that breaks the weak hydrogen bonds, opening up the two original strands. And as I mentioned before, not only does it bring in new nitrogen bases, but it proofreads. Okay, on to number 35. All right. So similarities between DNA and RNA. They're both NAs. They're both nucleic acids. They both can be found in the nucleus. Messenger RNA, well, there are three types of RNA. There's messenger RNA. I call them the three sisters. Messenger RNA is mRNA. Transfer RNA is tRNA. Ribosomal RNA is RNA. Messenger RNA actually gets produced, made in the nucleus, basically uses DNA as a template to take DNA's instructions from the nucleus. It leaves the nucleus through a nuclear pore, and it goes out to the ribosome where proteins are made. So messenger RNA can be found in the nucleus. DNA cannot leave the nucleus. Both made of nucleotide monomers. They both have adenine, cytosine, and guanine. Differences: DNA is double-stranded, RNA is single. DNA can only be in the nucleus. RNA can be in the nucleus and in the cytoplasm. Remember, the cytoplasm is all of the space outside of the nucleus, all the way to the cell membrane. So it's all that open space outside of the nucleus. DNA, the sugar is deoxyribose. RNA, the sugar is ribose. And DNA has thymine, whereas RNA has uracil instead of thymine. And as you remember from the movie, another characteristic is RNA is actually involved. Remember the RNA primer? RNA is actually involved in DNA replication. And that is one of the fundamental reasons that many scientists believe that RNA was the first nucleic acid on the planet, and then maybe RNA was the genetic material prior to the evolution of DNA, if you will. Getting ready to hop ahead. DNA replication is a process where a single strand of DNA has all the genetic blank necessary to reconstruct another chain of DNA. If you can't remember, you might want to take a look back at number, what does that say? 29. Yeah, genetic information. DNA replication occurs at how many locations at the same time and how many directions until the entire chromosome has been copied? You might want to take a look at number 30. Yep, hundreds of locations, both directions or two directions. Uh-oh, sorry. Let's take just a moment. There we go. Save it. Okay, the main enzyme involved in DNA replication, what do we call that enzyme? It joins individual blanks to produce a DNA molecule, and then it does what to each new DNA strand? You might want to take a look at number 33 and 34. Yep, named DNA polymerase. It joins individual nucleotides, okay, to produce a DNA molecule, and then it proofreads each DNA strand. Nicely done. Okay, in prokaryotic cells, where do we find the DNA? Yep, they do not have a nucleus, therefore it is in the cytoplasm. First step in DNA replication, produce new strands, separate the strands, produce DNA polymerase or base pair. Yeah, you got to have the helicase enzyme separate the strands to open them both up as a template, so then DNA polymerase can start building the two new strands. But you got to separate the strands first. Okay, easiest way to base pair is start at the beginning. All right, if we got a GCG, that means it's going to be a CGC, and only one of the choices starts with CGC. Normally on a test, if you have 4 choices, I will have 2 that start out correct, and then one of them obviously will have a couple mistakes in it, and you would pick the one that doesn't have the mistakes in it. In addition to carrying out replication of DNA, the enzyme DNA polymerase also, what does it do? Oh, my goodness. Man. Sorry. Perfectionist. What does it do? DNA polymerase, other than building the new strands. Yeah, after it's done, it goes and proofreads. All right, ladies and gentlemen, that is it for these notes. Please make sure that you load them into the classroom on time, and we will see you in the Zoom.